Hello and Assalamu alaikum. Today's session is exploring biological databases, the backbone of bioinformatics. As we all know, the last session is the introduction of bioinformatics, in which we discuss about the definition of bioinformatics, we discuss about the scope of bioinformatics, we discuss about the limitation of bioinformatics. In today's session, we will discover, we will explore the biological databases that are actually the backbone of bioinformatics. Actually, the bioinformatics is completely supported by databases and more specifically the biological data. So let's start with our the slides. So the Albert Einstein told if you can't explain it simply, you don't understand it well enough. Okay, welcome back to the Exploring the Biological Databases workshop session. My name is Salita Ras, and I am a BS Genetics and Molecular Biology scholar at University of Health Sciences. Brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics, and evolution, and at the end, molecular diagnostics. With a keen interest in advancing scientific discovery, Ali is dedicated to inspiring others through engaging learning experiences and fostering a deep understanding of cutting-edge molecular biology techniques. Okay, the workshop aim. This session aim is understand the role of biological databases. Okay, we will discuss about the role of biological databases. Learn how they serve as the backbone of bioinformatics and computational biology. Familiarize with the key databases and tools. Okay, we will discuss about the key databases and the tools uh, used in bioinformatics. We will explore the NCBI. We will explore the UniProt. We will explore the PDB. We will explore the ensemble and more for genetic and molecular research. Bridge biology and computational science. Okay, it is the bridge between the biology and computation, science. Okay, and here we will make the bridge between them. Okay, we will develop the skills in our upcoming sessions in which we analyze the biological data using the bioinformatics tools. Okay, for example, we want to align the two sequences. We use BLAST. Okay, we use to form the phylogenetic trees. We use Mega. Okay, there are different types of tools used to analyze the biological data. Okay, the most important biological data at this time is NGS data, next generation sequencing data. We will explore the data analysis in our upcoming sessions and enhance the data-driven research skills. Okay, when we have a data, we can do the research. Okay, and this type of skills have a lot of money. Okay, they gain the hands-on experience in sequence retrieval, annotation, and structural analysis. The today's session is not about the all these points. It is actually eh, about the introduction of biological databases. We will explore the key databases and tools. Okay, but the next points will be discussed in the next sessions. Okay, you will gradually uh, grasp the bioinformatics. Okay, by step by step. Not if you have just want to do a little more experience in something, you have to do by step, not uh, like jumps. If you jump one to one, it will really hurt you and you will get nothing. Okay, so step by step is uh, a good decision. Prepare for advanced topics. Okay, we will discuss about the sequence analysis, the phylogenetics, and molecular modeling. Almost the 8 to 10 sessions are about this. Okay, empowering the next generation bioinformatics. Okay, this is actually the point, and empower the next generation of bioinformatics. Okay, introverted but willing to discuss the bioinformatics. I love this thought. Okay, now we will discuss about the something like the bioinformatics, and then we go to the databases. Okay, bioinformatics is rapidly growing. Okay, it's a rapidly growing field. Okay, that merges, that make a bridge between the biology, computer science, mathematics, statistics, AI, machine learning, and a lot more. Okay, to analyze and interpret the biological data. It plays a crucial role in modern research. Now, the almost the half of the research is depend on the mathematics. Okay, helping the scientists understand the genetic sequences, the protein structures, the evolutionary relationships by using the bioinformatics. It's a really a fascinating program. It's a really a fascinating side of the life sciences, that is bioinformatics. Okay, now why bioinformatics? We will have experimental researches, we have a different types of life sciences programs, we have different types of PhD doctors having a good knowledge about the virology, about the molecular diagnostics, about the genomics, about the proteomics, about the transcriptomics. Then why bioinformatics? Actually, bioinformatics is only depend on the data. And today, the data is very vast. Okay, due to that data, like the Human Genome Project that is almost done in 2002 to 2003, actually produce a lot of data that actually sequence our whole genome, the human genome, more specifically, the human genome, our genome. Okay, and that sequence of that genome produce a lot of data. Okay, and that data is produced, but to analyze and interpret, to analyze and interpret this data is very difficult. It's very difficult. Why? Because the data is very fast and the human knowledge, the human efforts do not surpass the computers. Okay? That is why we use the computer to analysis the biological data. This is actually the produce the knowledge. That is actually produce a system that is actually more efficient, more reliable. Traditional methods become inefficient. Bioinformatics tools provides tools and techniques to store, the retrieve, and analyze the maximum amount of data efficient. That is why we use this. Okay, now we start from the database. What is database? Actually, database is a computerized archive. It means it's a it's a collection. Okay? It's a collection of data. Okay? It is like a bag. It's like a shopping bag in which the different types of things are available. Okay? And we just put the all things into one bag. And that one bag is called database. That is used to store. We store the things in it. We organize the data. Okay, we organize like that data is about the DNA, and that data is about the protein, that data is about the RNA, that about data is about these alignments, that data is about the phylogenetics. It's just like that. So that the information can be retrieved easily via various search criteria. Okay, we use the different types of search criteria we will discuss later. The chief objective, the chief objective of the development of database is to organize the data. The data is vast, the data is immense, the data is uh, very large. So we have to organize the data so we can collect, we can retrieve the data easily. We organize the data in a set of structured records. We form the records. Okay, the records we form the records in which the raw data, in which the raw data is stored. The raw data is stored in the organized way. Okay, and these records enable the easy retrieval of the information. Okay, then we retrieve the information easily. Each record also called an entry. Okay, we will discuss step by step. You will know about the all the words we use in it. Should contain a number of fields. Okay, it means that it's like a data. It's a data that is available in a organized way. Okay, it means that this data is organized and now it becomes record. Okay, now it becomes record. And one in records, there are different types of entries available. Okay, in which the number of different fields in which the entries are stored. Okay, it means that now the data is stored in the organized way and can be retrieved easily. And we can analyze it, analyze and interpret the data easily. Okay, to retrieve a particular record from the database, a user can specify a particular piece of information called an value. For example, we want to, for example, this is one, this is two, this is three, this is four, and so on. Okay. Now we want to analyze the particular field of the records. That is, for example, is the two. Okay. Now we use a piece of information like two. Okay, and we search it. Okay, we click on it and we search this two. That that entry, that is that field of the entry available in the record will be shown to us. Not the old records will be shown to us. It will become very very difficult to analyze the data. Okay, just one field will be in front of us, and we analyze that data. Okay, that can be done by using the value. We use the value. The value is the value we use is 2. Okay, to be find in a particular field and expect the computer to retrieve the whole data record. This process is called making. Okay, we give the value and the data available in it is retrieved into us. We just make a query. We just question and we just got the answer. Although data retrieval is the main purpose of those databases, it is one function to, to retrieve the data. Okay, the second function or the biological databases often have a higher level of the requirement. Okay, the second level is the higher level of the requirement, that is the knowledge discovery. Okay, it means that like we change the color. Okay, like that, it is the whole record we have. Okay, we just store in a way, but when we store, we got a new knowledge. Like that, the that data have some similarity in between. Okay, like this contains A, B, and C. Okay, and that data contains C, D, E. Okay, now what is actually this? That is actually E. What is similarity between A, B, C, and C, D, E? Yes, the C. Okay, that is actually the similarity between them. And for example, this is actually the genome of the mouse. Okay, that is, for example, we just assume it. This is actually the we can say it is actually the uh, genome of the mouse, and that is actually the genome of the human. Okay, now when we store the data in the organized way, we got that there is a similarity between the mouse and the human genome. Okay, that all is done by using the bioinformatics, by using the backbone of the bioinformatics, that is biological databases. By this, the identification, that identification of the connections between the pieces of information that were not known when the information was first entered. Okay, we entered the that field, we entered that field, we entered that field, we entered that field. We just doesn't know about what is actually the connections between that data. What is actually the there is actually the identification of the connections between that data. But when we put the data into the organized way, then there will be a knowledge discovery between the data. Will be overlaps with between the data, and we will discuss, we will check, we will interpret, we will analyze the data, that there is a connections between the data, and by this we do the research. That is why there is a lot of research potential in the bioinformatics, in the biological databases, and that is why the 50, more than 50% of the research of the biological research is only depend on the bioinformatics due to the knowledge discovery. Hope so you understand it. Okay, now we'll discuss about the types of databases. Okay, we will discuss about the types of biological databases later. Now we'll discuss about the actually the types of databases or the organization of the database. Okay, first of all, flat file databases. Okay, first of all, the flat file databases. almost the 80% of the biological databases are formed of flat file database. Okay, it's just a long text file that contains the many entries separated by delimiter. For example, it is it's like a data, and it's a, then there is a B, then there is a C, and then there is a delimiter. Okay, there is a long delimiter. Then there is a D, then there is a E, then there is a F. Okay, now see, now this is actually okay, then, then. Okay, now see, that overall is a record. Okay, and this is actually the field. Okay, and there is a there is actually the comma, and that one field, in the one field there is an entry. Okay, there are different entries. Okay, the relational databases. In the relational databases, we use the tabular forms of the data in the form of tables. Okay, we use the tables. That tables is called a relation. It was called the relation. Okay, in which the data is indexed. Okay, the data is indexed. It means that the when we open that database, we got that screen. Okay, like that screen, like that screen. There are three, like three records in only one screen. When we click on it, that will open in the new tab, and we got the next screen in which there is a different entries are available. Okay, it means there is the data is indexed. Okay, the programming used in it is SQL. Just just say the word SQL used in it. We will discuss later. Then the object-oriented databases. Okay, actually the problem of the both of the that databases is we don't that don't describe about the hierarchical relationships between the data items. Okay, we don't know which one is come first, and then what is the next, what is the second. But in the object-oriented databases, the complex hierarchical relationships can be analyzed. Okay, and the programming used in it is C++. Now we also use the Python, JavaScript. Okay, in it like that. That is flat plug. That is the data. Okay, that is the first is the name, then the state, then the course number, then the course name. Okay, and then there is a derivative. Okay, then again the name, the state, the course number, the course name. Okay, and this is the first student. That is the first student, the S1. That is the second student. That is the third student. And when and it is very computationally expensive. Okay, they have to go through the old data to find the something. Okay, but in the tabular form, like the relational databases, there is a connections between two. Like there is the name, like the Johann Smith. Okay, and the state is Texas. Okay, and that student is one. Okay, the student number is one, like that. Okay, there is a connections between them is one. When we talk about the Johann Smith, this is a there is a one student, that is student number one. The state is Texas, and the course number is that, and the course name is that. Okay, there is the actually the connections between the tables. That is why it is not as much flat file computationally expensive. And that is actually the object-oriented databases in which the databases are connected between each other by pointers. Okay, like that. There is a student, and there is a students like the student number one, student number two, student number three, and there is a course. Okay, and the course is connected with the course number. Okay, like that course number is the course number one, the biol 689, and the course name is bioinformatics. Okay, and then again the student is again the number one, and like that the tables are actually connected with each other, and there is a actually the hierarchical relationships between them. Okay, how the hierarchical relationships, like that. First of all, the student. Okay, then we comes up with the student name, then we talk about the student number. Okay, there is a one word student, and there is a two things we got is the student number and student name. Then there is a one word is the course, and we got two things, course number and course name. There is the hierarchical relationships between the data. Okay, now we'll talk about the biological databases. There are three different types of biological databases. First of all, the primary databases. It is very important databases. And then the secondary databases, which is also very important. And then the specialized databases, it is actually best for the people who are actually doing the specialization in something. Okay, like someone is doing their PhD in cancer, they will go to the cancer database. Okay, so it is actually specialized. In the primary databases, the old original database, the original biological data is stored in it. Okay, in which the original biological data is stored. Okay, this is actually the archives of the new sequence or the new structural data. Okay, that is actually submitted by the scientific community. Okay, you have to know that there is actually the primary databases in which the data is submitted by the scientists. Okay, the scientists do the research and got the do the research and got the result and submitted that data, that information into the primary databases, and the data is not organized in such a beautiful way, and not we can't analyze it very effectively. Okay, the different types of the biological primary databases is the GenBank, like the NCBI, and then the protein data bank like the PDB, in which the 3D in which the 3D structural in which the 3D structures of the proteins are stored. Okay. Now, the problem of the primary databases is that it is not computationally annotated. Okay. It is not computationally created. Okay. We have to create the data. We have to connect the different data into one something. Okay. Like there is a data is actually vastly separated, vastly spread into the whole room. Okay, but we have to store the data like uh, something is always into the red box. The second one is the green box. The third one is the yellow box. Okay, that type of the data then created. Okay, and now we want to just explore the red data, the red box. Okay, we just grab the red box. We don't touch the green or the yellow data box. Okay, that is actually the curation of the database, or sorry, of the data, and process the data. Okay, and in the secondary database, Okay, in actually secondary databases, we actually computationally processed or manually corrected the information. Okay, in which like the Swiss prot, like the protein information resources, the PIR, we actually manually create the data. Okay, in which like something with that in the primary databases, we store the data like the DNA, then RNA, then protein. It is actually the central dogma. Okay. In the primary databases, the DNA sequence, the RNA sequence, the protein sequencing is stored. Okay. But in the secondary databases, we will discuss about the the things available in the data, available in the DNA. Like there is actually actually available is the promoters. Promoters are available in it. Okay. Actually, the exons are available in it. The introns are available in the DNA sequence or in the RNA sequences. And there is a polyethyl available in the messenger RNA. There is a uh, different types of like the transcriptomics data are available. The different types of proteomics data is available in the DNA sequencing or RNA sequencing or protein sequencing. Okay, that will not be curated by the primary databases, so we have to do that curation by using the secondary database. And the specialized databases, we cater the particular research interest. Okay, in which the particularly we we are very specific. Okay, we are very specific in the specialized. Like maybe it is about the genetically diseases. Okay, genetic diseases database. It may be about the cancer database. It may be about the HIV. It may be about the molecular diagnostics. It may be about the mm, sorry, we may be about the breast cancers. Okay, there are different types of databases that are specifically cater the particular section across. Okay, this is the table in which the ASDB is the genome database for the Chinorhabditis elegans, the DDBJ, the data bank of uh, the DNA data bank of the Japan, the AMBL, the European Molecular Biology Laboratory, the ENTRAS is actually the NCBI portal, variety of biological databases, the XPACY, the Flybase, the FSSP, the GenBank, the HIV database, the microarray, the OMIM, the PIR, the PubMed, the ribosomal, the SRS, the SWISPROT, the TAR. You can explore it by using that URL. Okay, and that is actually the brief summary. Okay, now if you have any questions and in your mind, you can check it out by using comments me, or you can also follow me on or make a connection on the LinkedIn, subscribe on the YouTube. You can also email me. You can also follow the channel. And if you have any questions, you can message me personally. I will definitely answer you. So thank you for being part of this wonderful journey. Follow for further virtues on sound announcements and lot more. And also subscribe, like, and comment about if you have any query. And please turn on the notification. So if you want to uh, follow the next sessions, you will definitely notify about it. Thank you for today's session. In the next session, we will discuss about the NCBI, the UniProt, the Ensemble, and the different types of databases, and we will also discuss about the sequence analysis, the blast, how will we use it. This is very if you fascinating the bioinformatics. It is very beautiful. It is very beautiful. If you are not fascinating, when you do these sessions, you will definitely fascinate the bioinformatics. Thank you for today. And please pray for me. Happy Ramadan to all of you and Allah Hafiz.